Do you know what is glycolysis? It is the process in which glucose is broken down to produce energy that takes place in the cytoplasm of a cell and does not require oxygen. Besides, it occurs in both aerobic and anaerobic organisms. So, this is the pathway of glycolysis. There are 10 stages which will be discussed further. So, let's get started.
The first step in glycolysis is the conversion of glucose into glucose 6-phosphate. Here, the glucose ring is phosphorylated by the adding of a phosphate group derived from an ATP. This means that one molecule of ATP is consumed during this phase. The phosphorylation reaction is catalyzed by the enzyme hexokinase that catalyzes the phosphorylation of many 6-membered glucose-like ring structures. Glucose 6-phosphate is formed because the 6th carbon of the glucose acquires the phosphate group. The second step of glycolysis involves the conversion of glucose 6-phosphate to fructose 6-phosphate. This reaction occurs with the help of the enzyme phosphoglucose isomerase. As the name of the enzyme suggests, this reaction involves an isomerization reaction. The reaction involves the rearrangement of the carbon-oxygen bond to transform the 6-membered ring into a 5-membered ring. Next, step 3 is phosphorylation of fructose 6-phosphate. Fructose 6-phosphate is further phosphorylated to fructose 1,6-bisphosphate, which is catalyzed by phosphofructokinase. A phosphate group is transferred from ATP to fructose 6-phosphate and becomes fructose 1,6-bisphosphate. 1 ATP is utilized in this step. Then, in step 4, cleavage of fructose 1,6-bisphosphate occurs. The enzyme aldolase splits fructose 1,6-bisphosphate into a ketone and an aldehyde molecule. These sugars, dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, G3P, are isomers to each other. The reaction is reversible. Step 5. Dihydroxyacetone phosphate formed during aldolase reaction is then further converted into the second glyceraldehyde 3-phosphate with the help of triose phosphate isomerase enzyme. Step 6 is the first reaction of energy-yielding step. During this phase, glyceraldehyde 3-phosphate is oxidized by the coenzyme nicotinamide adenine dinucleotide NAD, which then is phosphorylated by the addition of phosphate group forming 1,3-bisphosphoglycerate and hydrogen ion. Besides, the enzyme that catalyzes this reaction is glyceraldehyde 3-phosphate dehydrogenase. Both glyceraldehyde 3-phosphate are involved in this reaction, thus producing a total of 2,1,3-bisphosphoglycerate. Step 7. In stage 2 of glycolysis, ATP is produced as a result of the loss of a phosphate group from 1,3-bisphosphoglycerate. With this step, we have cancelled out the first 2 molecules of ATP that we used, leaving us with just 0 ATP molecules. Step 8. A mutase is an enzyme that catalyzes the transfer of a functional group from one position on a molecule to another. This step involves a simple rearrangement of the position of the phosphate group on the 3-phosphoglycerate molecule, making it 2 phosphoglycerates. Step 9. Dehydration of 2-phosphoglycerate. This step involves the conversion of 2-phosphoglycerate to phosphoenol pyruvate, PEP. The reaction is catalyzed by the enzyme enolase. The enzyme removes a molecule of water from 2-phosphoglycerate to form phosphoenol pyruvate. Step 10. Transfer of phosphoryl group from ATP to ADP. In this step, phosphoenol pyruvate were converted into pyruvate with the help of enzyme kinase. As the enzyme name suggests, this reaction involves the transfer of phosphate group. The phosphate group attached to the second carbon of the PEP will be transferred to the molecule ADP. Then, a molecule of pyruvate and ATP will be generated.
Let's look at the net reaction of glycolysis. We have one molecule of glucose that's split into 2 molecules of pyruvate. During the latter stage of glycolysis, 4 ATP molecules are synthesized using the energy given off during the reaction. Thus, 4 molecules of ATP are synthesized and 2 ATP molecules are used during glycolysis for the net gain of 2 ATP molecules. 2 molecules of inorganic phosphate are utilized in step 6, and 2 NAD are reduced into 2 molecules of NADH.